Hello, and welcome to Plants in Python. My name is Bob Van Buren, and I'm an assistant professor in the Department of Horticulture at Michigan State University. And in today's lesson, we will talk about variant discovery and quantifying genetic variation in plants. And so far in these lessons, we've talked about how to assemble a genome using raw data, how to annotate and compare genomes between species, and what components make up a genome. And we've also talked about how to quantify some element of activity within a plant with an emphasis on gene expression or RNA-seq. And so next step in this lesson, we're going to be talking about how to quantify genetic variation between individuals within a species. And so for most of our crops, there's tremendous genetic variation. So here's an example of tomato. So these are just a representative of a number of different tomato accessions. So we have here at the top some of these, what you would refer to as like heirloom varieties. So these are things that you might find at a farmer's market. These are a little bit older. They tend to be a little bit lower yielding. We have some down here, more commercial varieties. So these are what you would see in tomato processing, and some close to wild species as well. And so if we're doing breeding or quantitative genetics, we might want to know, how do these individuals differ from each other, and how can we use that variation for some applied research. So instead of looking across the entire genetic diversity of a species, maybe we could have two different individuals that differ in a trait of interest and ask, what is the genetic basis of this trait? So in this example, we're using apple skin color, and so we have these green fruits and these red fruits, and so we might want to know, what's the underlying genetic basis of this trait? And so we could make a cross here with our parent 1, which is a red apple, and parent 2, which is a green apple. I'll use this example a little bit later in this lesson. We could make an F1 or an offspring, and maybe this offspring has some combination of these traits. And then we could make some advanced population where we keep selfing these individuals, and then we get this 8th generation of plants here, where we have some that match the red parent's phenotype, some that match the green parent phenotype, and some in between. And so we might want to say, okay, we have all this genetic variation. What's the underlying differences between these two individuals, and can we map or identify a variant that is causing this difference? So this is an example from my own research in pineapple. And so this is similar to the apple. This is an F2. So these are a wild pineapple crossed with one of the leading cultivars, and then that offspring is selfed. And then these are just some of the different pineapples that come from that. So you can see there's huge variation in skin color, in size, in shape, even in leaf color. And then if you were to taste these, they have massive variation in different traits. And so the question is, how do we, so now that we have these phenotypic differences, how do we identify and quantify this genetic variation? Like so many of the different things we're going to talk about in this course, we could spend a whole semester talking about variant detection and some of the downstream analyses that we can do. You could take a whole course on quantitative genetics or plant breeding. You could take a course on population genetics or evolutionary biology. But in today's lesson, I kind of just want to provide a brief overview of the way that we do these analyses and the type of downstream things that you can do. And so the plant biology learning objectives are listed here. And so the first is to understand the principles of realignment and variant detection. So how we can use Illumina data to identify differences between individuals in a population or individual accessions. We want to understand the downstream applications and analyses associated with variant calling. So once we have our matrix of differences between our reference and whatever individuals we're looking at, what types of analyses can we run, and what are the limitations and the strengths? We want to learn the distinction between small and large scale variants. And so typically maybe before 2018, most of the variants we would identify are going to be in the range of a single nucleotide or a couple base pairs. But now with the decrease in cost and increase in quality of long-read data, we can identify all of the large-scale structural variants between individuals, and that's very powerful. And then at the very end, we'll introduce the concept of pangenomics and some of the applications that we can have with that. And so again, we're going to kind of skim the surface here, but if you're interested, there are a lot more in-depth lessons or entire courses that you could take on any one of these topics. And so there are generally two ways we can go about identifying variants between our individual of interest and a reference genome. And so the first one that we're going to talk about, this is by far the cheapest option, and historically it was the one that was done most commonly until maybe 2018, 2019. And so this is using short-read Illumina data and aligning it to a reference genome and identifying differences. And so here we have it as this example. We have our reference genome on top. So this would be a small chunk of this reference genome. And we would have our Illumina paired end data here, where we have this insert of maybe 500 or 600 base pairs. And then with paired end, we would sequence a little bit from the start of this fragment, and then a little bit of the end of the fragment as well. And so we'd have about 100 base pairs here and about 100 base pairs here. And so we would take these reads and then use a program such as BWA or Bowtie2. These are probably the most common, most powerful aligners. We would run a computationally intensive alignment and see exactly where those align in the reference genome. And then this is kind of an example here. Again, if we're doing shotgun sequencing, these reads are going to be randomly in the genome. And you can see here we have pretty high coverage. And then we can look at where these reads are aligning and then run a variant detection tool where we'll go base pair by base pair of the genome and see if there are any differences between the reads that we're aligning and the reference genome. So this is kind of the way it was done traditionally, and this is the most, at least cost intensive approach. And so in general for a plant genome, if you were looking at something that was human sized or a little bit less, you can resequence an individual for a couple $100 US. So it's relatively inexpensive to do variant detection using this approach. And so this is kind of an example of what the data might look like. And so here we would have our individual chunk of the genome here, and then each one of these along here would be a read, and then these are also colored by the base pairs, and so the green, purple, orange, and red correspond to a different nucleotide. And here you can see this is pretty high coverage where we might have hundreds of reads that are aligning to each base pair in the genome. But it's important to note here as well that this is somewhat uneven, right? So we have a lot of coverage here, but a little bit less coverage down here. And so the question is, how much data do we need? And this really depends on the downstream application that you're doing. So we can kind of start from the bottom here of the highest coverage, and so this would be really important for clinical data. So if you're working with human disease-associated types of analyses, you might want really high coverage. So that would be on average 10 to 100x. And so if you remember back a couple lessons, what this means is that every base pair in the genome would be sequenced on average 10 to 100 times. And so I haven't gone through and counted this, but this would probably be equivalent to about 100x coverage. And so the benefit here is the higher the coverage of the data that you have, the more accurate your read calling or your variant calling is going to be. And so we can also have relatively low coverage data. So we would say this is in the order of 2 to 10x. And so if you do the math, like if you only had an average 2x coverage of the genome, there would probably be about 10% of the genome where you had no reads for that. So that can be kind of problematic. And then if you go up to 10x, you should get 99+% of the genome where you have at least one read aligning to it. And then you'll still have some parts of the genome where you don't have any data. And then you can also do a skim sequencing approach where you would only generate on average 0.5 to 2x coverage of the genome. So in this case, you would have probably half or more of the genome where you didn't have any data coverage. But this is really powerful if you only need a small number of variants. So, between any individual and the reference, there can be millions or tens of millions of differences, but maybe you only need to capture a couple thousand of those differences to do the downstream analyses that you want. So the coverage really depends on your question, but I would say in general in plants, we'd probably shoot for this between low and high coverage where we would get maybe on the low end of 10 or 20x coverage and the high end of 50x coverage. And that ensures that we're getting all of the variants that we can. And also this depends too on polyploidy. So if you have a polyploid individual, you want higher coverage because you want to ensure that each of your individual base pairs has good representation from each of your haplotypes or each of your alleles. So it really just depends. So there are a number of different types of variants that we can identify. And the first one, and this is probably the most common one that you would encounter in a lot of downstream analyses, this is called a single nucleotide polymorphism. And so this would be it could be, in this case, this is a SNP, we would call that a SNP of an A turning into a C. So it's just going to be a single base pair where one base pair is substituted with another. And so in this example here, we have our reference genome here, and we can see at this position the reference is an A, and then each one of these is going to be an Illumina read that is aligned to that reference. And you can see here that all these reads except for this one are C. And so what we would say here is that this individual has a SNP, or a single nucleotide polymorphism, from an A to a C, where the reference line is an A, and the accession, or the individual we've sequenced, is going to be a C. And so these are the most common. They're the easiest to quantify. They tend to be very accurate. And typically we would use these in some sort of character matrix, which we'll talk about a little bit later in this lesson. We can also have hemizygous sites. And so for diploid plants, we would have 2 copies of each allele. So one that would be inherited from the mom and one that would be inherited from the dad. And so these are a couple examples of hemizygous alleles. And so for instance here, we have the reference line is a T, and then in our individual we have some reads that are T and then some reads that are C. And so this would be hemizygous or heterozygous of T/C. This base pair here, the reference is a C, and we can see here that some of our reads are C's and some are T's. And so again, maybe the copy from parent A would be a C, and then parent B would be a T. And then you can see a couple of hemizygous sites here as well. So you can have homozygous, where it's going to be both alleles are different from the reference, or you can have a heterozygous or hemizygous site where you have one allele that matches the reference and one allele that's different, or even 2 alleles that are different than the reference. That happens as well. So these are the 2 types of variants that we would see for single nucleotide polymorphisms. So we can also have slightly different variants called indels, or small-scale insertions or deletions. And so this would be a case where you would have a small chunk of DNA that's found in the reference that is not found in your resequenced individuals. Or it could be a couple base pairs that are found in your resequenced individual, but that aren't found in the reference. And so in this case here, we have the reference at this position is a C, and then most of our reads here this star denotes that there's no base pair there. So this would be a deletion, where the reference line has a C, and our individual has no sequence there. And what's kind of interesting here is that you'll see that there's a C here, and what this probably is this is an error in the alignment where this C probably should be moved over to one base pair here. But because it's a deletion, we can't accurately quantify that. And so in general, Illumina data is able to identify pretty accurately small-scale insertions or deletions in the range of 10 to 20 base pairs, but then when you go beyond that, because of the limitations with the read technology, we can't accurately identify those. And so in general, these small-scale indels tend to be a little bit lower confidence. You can have read alignment issues at these positions like you're seeing here. So in general, when we're doing a lot of downstream analyses, at least using Illumina data, we are not going to be using indels for a lot of things. And then what's also kind of interesting to note here is that at this position here, you can see all the reads are a T, and then we have the single read here that's a G. And so this would correspond to an actual error in the Illumina read. And this is something that we also have to deal with a lot of this data. Is like, in general, the Illumina data is like 99% 99.5% accurate, but every once in a while you get an error like this. And so when we're doing filtering, we need to have strict criteria so that we wouldn't have this single G here be called a polymorphism. So that's really important when we're doing our downstream analyses. And so those are kind of small scale variants, so either a single nucleotide that switches from one base pair to another, or a little bit larger variants of maybe 10 or 20 base pairs. But plant genomes are pretty dynamic in general, and you can have, if you remember back to the second lesson when we're talking about the elements of the genome, we can have repetitive elements that move around, that duplicate themselves, that move other sequences around, and so you can have thousands or tens of thousands of these long terminal repeat retrotransposons or retrotransposons in general that are going to be different between individuals. And so a lot of the differences that we would identify between individuals will not be captured using Illumina short-read data. And so we can identify these complex variants, and we would use this in like a pangenome approach, which we'll talk about at the very end of this lesson, where maybe we would have this large-scale deletion, where we would have this reference is going to be at the top here in all these examples, and then here is our sample. So this could be a different variety of our crop species. It could be a wild species. Maybe it's a mapping population of a biparental cross or something like that. And in this case, we have reference has this large chunk of DNA that is not found in our sample, so this would be a deletion. We could have a novel sequence insertion here, where we have our reference has nothing, and then our sample has this big chunk of DNA. So this could be a gene, it could be some weird horizontal transfer from another species, or most commonly this is going to be a transposon that would insert. We could have an interspersed duplication, where we would maybe have in our reference this one blue chunk here, maybe this is a gene, a transposon, and then in our sample we have this chunk has been duplicated somewhere else. We could have a tandem duplication. This is very common in plants, where our reference would have a single copy of this gene or this chunk of DNA, and then the sample would have 2 or more copies. So this happens a lot during meiosis, where if you have uneven crossing over, you can very easily delete or duplicate chunks of DNA. And so in any plant genome, there are on the order of thousands or tens of thousands of tandem gene duplications. And so these have really important biological functions, so we need to capture those. You could have an inversion, where this chunk of DNA is maybe going in this direction here, left to right, in our reference, and then in our sample it's going right to left. And so that can also be an issue during meiosis. We could have a translocation, where we have a big chunk of DNA that moves from one region of a chromosome or one chromosome to another chromosome. And we can have weird complex things, like maybe we have a deletion and an inversion, or insertion and a deletion. Or in this case, we have maybe this like inverted sequence that gets deleted or moved somewhere else, and then we have an insertion or something. And so when we're looking at single nucleotide variants, we're looking at a very simplified picture of what the genome looks like, right? We're just looking at, here's this base pair, it has a C, the other genome has a T, but in reality, there are tens of thousands of these more complicated differences that we don't necessarily capture with short-read data. And these can have really important biological implications. So I would say in general, most of the evolutionary innovation in plants is because of these types of things, rather than a single nucleotide that switches an amino acid to something else, and then that causes a shift in a protein. So a lot of domestication type genes, a lot of resilience type genes are caused by these complex variants. And so at the very end, we'll talk about how we can identify these and kind of where the field is moving. So now we've talked about the different types of variants, the question is, how do we identify them? And so like anything in bioinformatics, there are a number of different softwares that we can use to do this. The general principle with this is that we have our sort of pre-processing, where we would take our raw reads and align them to a genome and do some quality control stuff. We would have our variant discovery, where we take those aligned reads and we look at the reference and see what's different. And then we also have our kind of cleanup quality control, where we filter our downstream variants. And so the most common program to do this is GATK, or the Genome Analysis Toolkit. And so this was developed by the Broad Institute, and this is currently probably the gold standard variant identification program. There are a couple others, but this is probably the one that you're most likely to encounter, and at least in my opinion, I think this gives the highest quality data for using Illumina data. And so there are a lot of steps to this pipeline. We won't run it in this course because it can take days or weeks. It's very computationally intensive, depending on the number of individuals you have. And that's because you have your pre-processing where you're aligning all the reads against the genome. And then when you're actually discovering the variants, GATK is very sophisticated and does a really good job, a lot of quality control metrics. And so it takes a lot of computational power of searching along individual reads, identifying variants, filtering them, joining them, things like that. So we won't run this in this course, but we'll talk about the conceptual framework. And so the first step is aligning the reads to the genome. And so we would take our raw reads, these would be raw Illumina reads, we would quality filter them, get rid of any adapters, any low quality base pairs. We could use a couple of different programs to actually map these reads to our reference genome. So the most common, as we talked about, are BWA and Bowtie2. And so what these will produce is a BAM file that has the read itself and then the exact positions that it aligns to the genome and any differences between the reference and the genome line. We would do some sort of filtering where we would mark any PCR duplicates, remove them using Picard tools, and then we would kind of do some filtering and some renaming the reads and things. And you might also do a base recalibration where you would just make sure that there aren't any biases in your samples. And then we would take this cleaned up, sorted, processed BAM file of where the reads are aligning, sorted by position in the genome, and then we would take that and bring that into the program GATK. And so we would call those like analysis ready reads. And then so what GATK will do is it will run one of a couple different variant detection algorithms where it will take this file of analysis ready reads and it will go base pair by base pair through the genome and say, okay, at this base pair we have in the BAM file maybe 10 or 20 reads that show a difference. It will do some local realignment if there's indels and things, and then it will look at the genotype calls and how many individual reads map the reference, how many don't, and then it will produce this genotype likelihood here. And this is just basically a quality score or how confident GATK is that there isn't a variant. And then we would run this process on each individual that we would have. So these could be individual accessions, they could be individual, like in a mapping population, like different siblings. We would merge those together and then do joint genotyping where we would compare the putative variants that were identified in each individual. And then we can take that raw data set, filter different variants. So maybe we want to remove the indels because they're not as high quality. Maybe we want to remove some of the lower confidence SNPs where GATK wasn't super confident that those were real. Maybe we would want to filter down. So if we have 20 million variants, maybe we want to filter based on their position in the genome or their linkage disequilibrium, which we won't talk about that. And then you can kind of go into your various analyses. You could go back and rerun the pipeline. You could run it against a standard set or something like that. So this is a very intensive process, but in general, you have to make a lot of decisions. So maybe 98% of your SNPs are going to be high quality, but the question is how many do you need and what thresholds are really important to you. So is it better to have more slightly lower quality variants or a refined set of really high quality variants? And that depends on the downstream analyses. So we won't run the pipeline in this course, but if you want to learn more about this, there are tons of resources out there on GATK. And so once we have our final genotype matrix, what does it look like? And so we have this file, the most common file that you'll see with variants is this VCF file, or the variant call format. And so in this example, I'm going to use the apple example again, where our reference, maybe this is a red apple, would have the genotype of an AA, and then the alternative genotype. Maybe this green apple would be CC. And so we code this using binary character traits. And so when we're doing downstream analyses, we can't really put A's, C's, T's, and G's into a character matrix for hierarchical clustering or PCA or something. We can classify them as zeros and ones. And so a 0 would be basically the reference, and a 1 would be the alternate. And so if we have a homozygous reference, it would be for a diploid AA or A/A, and that would be a 0/0. If we have a hemizygous or heterozygous, where the individual has one allele from the reference and one from the alternative, we would give this a 0 for the A and a 1 for the C, and so this would be a genotype of 0/1. And then if it's both alternates, so allele A is a C and the allele B is also a C, it will be 1/1. So this is kind of the nomenclature here. And so this is what the actual genotype format looks like. There's a lot of different things to point out here. And so this is just a VCF file. It's a tab delimited file. I've just imported it into Excel to kind of show you here. So we have our first lines. These are denoted by hashtags. And these are just kind of tells you the version of VCF, the file date, the reference, the alleles, all kinds of just basic information about what was run. And then in green here, or sorry, in red here, we have a couple different important fields. And so the first column is going to be the chromosome. This could be the number of chromosome. It could be chromosome 1, 2, 3. It could be contig or scaffold 1, 2, 3. We have our position in the chromosome. And so like this would be basically base pair 45,794,806. An ID, so this is if that genotype or if that SNP or variant has an ID, most of them don't. The reference here would be, in this case, it would be a C. So the alternative here is an indel, or in this case, it's an insertion, where the reference is a C, and then the alternate has a C and a T. A quality score, which we don't always have that. If it passes or fails, you're probably going to only have the passes here. And then this next group is various information. We won't really talk about that too much. And then the part that we care about most is going to be the format. So this tells you the different metrics that it's putting in here. So this is going to be the basic three that we care about are the first one is the GT or the genotype. And that's going to be our 00, 01, or 11. The depth, this is the number of reads that we have at that position. That's really important because we can filter based on that. And then the allele frequency of how much are the alternate and how much are the reference. And so for example, in this base pair here, and then we would have a column for each individual. And so this is S1 or this is sample 1, but if you were resequencing 100 samples, you would have 100 columns where each column was the genotypes for that individual. And then here we would have this 0/1. So the 0 would be the reference of a C. The 1 allele would be the alternate. So this is a hemizygous site. The depth here is 265x coverage. And then the genotype allele frequency is 0.51, which means roughly half the reads are the reference and half are the alternative. And then we can go down, and for here example, we have, this is a 1-/1. So in this case, both the alleles are a C, whereas the reference is a T. And then the allele frequency there is 1.0. So that's basically all the reads were an alternative. And so those are really good. We also got some weird stuff like allele frequencies of 0.14 or 0.11. These could be, we would expect it to be basically 0.5 or 1. I don't know what's going on here. These could be low quality ones. So then we would want to filter this. And what's really important is for downstream analyses, we want this genotype matrix here, and we can use this to identify differences between individuals for maybe a genome GWAS or genome-wide association study, or maybe some quantitative genetics, or we can use this and convert it into a binary character matrix and look at differences between individuals, which we'll talk about in the next slide. And so again, we can't really run statistics on this using a SNP matrix of A's, T's, C's, and G's. So we can turn this into a binary character matrix. And so here we would take our VCF file and then we would convert it just to the A's, T's, C's, and G's. And so for each row we have a different SNP. So this would be SNP 1 through SNP 6. And then this is the genotype. Each column is the genotype for that SNP in each of these individuals. So this would be individual 1, 2, 3, 4. And so what we can do is turn, again using this sort of nomenclature where we have, in this case, a homozygous reference would be a 0, a heterozygous of the reference and alternative would be a 1, and then a homozygous allele with 2 alternative alleles, so in this case, let's say C/C, would be coded as a 2. And so we use this 0s, 1s, and 2s, and this will denote the genotypes. And so we could convert this file here from A's, T's, C's, and G's to a single number for each individual, where a 0 would be homozygous reference, a 1 would be heterozygous, and then a 2 would be homozygous alternative. And then we can use this character matrix, in this case it would be 0s, 1s, and 2s, to run downstream statistics. And so there are a number of different approaches that we can do here. So we can do a principal component analysis, which we will actually run in the notebook associated with this video. We can also run a hierarchical clustering, which we will also do in this video. And so the way this works is, let's say we have this grouping of data here where we have these points A, B, C, D, E, and F, and this is sort of their dimensional space here. And so if we want to cluster these, we could look at the Euclidean distance between all these individuals, and we could say, okay, on this distance, E and F are very close to each other, A and B are close to each other, and then we have kind of this 2 grouping here, where A and B are over here, and C, D, E, and F are over here, and then we can look at the Euclidean distance of each of these individual points against one another, and we can kind of do this in a hierarchical way, and we can build this dendrogram here, where this is a good representation of our space here, where we have sort of going from the top here, we have this 2 grouping that it's producing of A and B over here, and C, D, E, and F as this big group over here. And then we have A and B are close to each other, and then within this group of C, D, E, and F, we have E and F are sort of pairs, and then E and F are closest to D here, so the Euclidean distance would be a little bit less here. And then C is a little bit further away from all 3 of these. And then A and B are the furthest away. So we can build this, run this hierarchical clustering where we kind of build from the ground up and produce this dendrogram of differences. And in this case, we're kind of showing this just using this 2D space, but we can also use our character matrix here with each of our individuals and then our genotypes to build a dendrogram with using hierarchical clustering to see how individuals relate to each other. So this is an example of some of my research. So this is a paper that was published a couple years ago on pineapple domestication. And so here, this is showing basically a hierarchical clustering of different pineapple cultivars and their wild relatives. And so this is probably the most common pineapple that you've encountered in the grocery store, at least here in the US. So these are the kind of cayenne group cultivars. So they're really large, they have yellow flesh, very sweet. And then we have 2 other groups that you would see, these most commonly in Southeast Asia and in South and Central America. So these queen varieties, which are really small, kind of individual sized, they're very, very sweet. And then this Singapore Spanish, which is also very small. And then we also have these wild relatives of pineapple, these different subspecies that look kind of like pineapple, but they're a little bit different. And the question was, which of these subspecies was pineapple domesticated from? And so what we did was we resequenced a couple hundred individuals. We identified tens of millions of SNPs or variants between the individuals in our reference genome. And then we used a subset of those SNPs and ran a hierarchical clustering. And then what we can see here is we have all of our cultivar groups here in yellow, blue, and purple. And then these are most closely associated with this subspecies rectifolius. And then we have these other outgroups here that are more distantly related. And so we can use this sort of this relations here to say that domesticated pineapple is most closely related to this wild species here. We can also run a principal component analysis, so if you think back to the last notebook, and we can use this character matrix as an input for that. We can filter it and then transform it and run a PCA or some other type of dimensionality reduction. And again here what we can see is we have all of our cultivars down here, and this clustered really closely together, so pineapple has kind of a narrow domestication history. And then we have here in red, these are the wild species, Rectifolius in red, and then these are some of the other outgroups here, this Bracteatus, and then this Microstachys here. And so again, what this is showing is that when we do dimensionality reduction, all of our cultivars are most closely related to this subgroup here. And so we can use this combination of dimensionality reduction and hierarchical clustering to look at the relationship between different accessions or different individuals. Here's another example using tomato, and this is somewhat similar but a little bit different. So you could take whole courses on quantitative genetics, but I'm just going to mention this in this one brief slide here. And so this was a paper from 2014, and the authors identified variants across 360 tomato accessions. These were some domesticated, wild, and land races, which are kind of in between domesticated and wild. And then this is a results from a genome-wide association study. And so basically what you would have is for a simple qualitative trait, you might have skin color of pink tomatoes versus, pink tomatoes over here versus red tomatoes over here. And then you would basically say, okay, we have this list of a couple million variants. Which ones are associated with cultivars that have red flesh and which ones have pink flesh? And then you run a statistical test on each one of those SNPs. And what they basically found was this single region of this chromosome, and each base pair here has a LOD score. That's basically a likelihood that each one of these SNPs is associated with red or pink flesh color. And the authors identified a single SNP here that had a super high LOD score, so very confident that this is associated with this fruit color. And they allowed them to identify a single SNP that distinguished red and pink fruit during tomato domestication. And so you can use this group of variants to identify differences that are associated with your trait of interest. So this is a really simple example, a qualitative example of red versus pink flesh, but it could be quantitative genetics data, so like maybe looking at yield or nutrition or stress tolerance, something that occurs along a continuum. And so you can use this massive character matrix of data from individuals and use this for quantitative genetics as well. So again, you could spend whole courses talking about this, but this is just an example of what we can do with this resequenced data. Okay, so lastly in this notebook, I want to talk a little bit about where the field is moving in the area of pangenomics. And so what we've talked about before was looking at these single nucleotide variants or these small indels, insertions and deletions, but the problem is that they don't capture most of the true genomic variation between individuals. So they're really good at capturing the single base pair differences, but they're not very good at capturing any large structural changes that occur in individuals. And these are really common either because of errors during meiosis, because of movement of transposons or other features. And a lot of these differences tend to be associated with important agricultural traits. And so we refer to a pangenome as kind of the total representation of all of the differences between a reference and all the other accessions that we've sequenced. And so the way that it works here is very similar to short read assembly, where we have our reference here, and then we align our reads to it. And so in this case, instead of just aligning them and identifying variants, we're aligning very large reads. So these would be like third generation sequencing, so single molecule based reads at a couple kb or maybe even tens of kb as well. We would align these to a reference genome, and then we would take any unaligned reads, we would assemble those together, and then we would see where they go in the reference. And so in this case here in red, we see these reads don't align anywhere into the reference genome because that's a chunk of DNA that's not found in the reference. We can take those separately, assemble them using this kind of iterative process, and then build this pangenome reference where we have everything that's found in the original reference, and then some of these big chunks that are not found in the reference. And we can take all of these de novo assembled genomes, so we would generate a de novo assembly, either using this iterative approach or just entirely independently, where we take our raw reads and assemble them. We can align them against each other, so this is very similar to what we did in the second lesson. And then you can look for any differences that are found between the reference and the other genomes. And so we would distinguish these two different categories into core and dispensable genome regions. And so the core regions are going to be found in all of our reference genomes. And so if you think about this for rice, for instance, these would be like all the chunks that are found in every rice variety. And these are kind of like what's essential to make rice at a foundational level. And then we would have the dispensable genome, and these are just chunks of DNA that are found in only one or a couple references. And so in this example here, we have these kind of conserved blue chunks that are found in every individual, and then maybe we would have these red chunks where this piece of DNA is expanded in this individual. Maybe this red piece of DNA is inverted, where it's in one orientation in individual 1, and then that flips around in the opposite orientation in individual 2. And then maybe this entire chunk of DNA is not found at all in this third individual. Maybe these kind of move around a little bit where there's some smaller sequences here. Again, maybe we have this big chunk that's found in this individual, but not in this individual, and this chunk is smaller in the third individual. And then what we end up getting is we take all these differences and we build a pangenome graph where we have all of our core genome is going to be a single collapsed region, and anywhere that we have a difference, we would have these multiple paths that could correspond to it. And so here, in this example, this chunk of DNA is a little bit bigger than this chunk, and so maybe this is one piece of DNA where it could be this sequence or it could be this one. Maybe in this case, it could be the chunk of DNA in one orientation, the chunk of DNA in a different orientation, and maybe this entirely new fragment. And this is a much better representation of what a genome actually looks like. And so because genomes are highly dynamic in plants, we have some chunks that are represented in some accessions and not another. This is kind of where the field is moving to, instead of having a reference genome as a single FASTA file that just goes from start to end, we would have this more complex graph structure where all the possible variants that we've identified between individuals can be found in this more complex graph. And so what's kind of interesting is that they found in some genomes, and we'll talk about this in the next slide, that up to half the genome or more can be considered dispensable or variable between accessions. And so if you're using a single reference genome in your studies, maybe you're missing a lot of really important biological context. And so that's just one example I want to talk about from rice. So this paper was published earlier this year in summer 2021, and similar studies are coming out for all kinds of species. And so in this paper, the authors did de novo genome assembly for 31 or 33 individual rice accessions. And so you probably don't recognize a lot of these names, but here's one, basmati. You're probably familiar with that, different rice. But these are all different groups of rices. They are different functional classes. They have obviously very different phenotypes. And so what they did was they assembled a de novo reference genome for each of these individuals, and then they compared them to the current rice reference genome. And then so each one of these, so these are corresponding to insertions in green and deletions in red. And what's interesting is that each rice variety has at least 10,000, or in many cases, many more, large-scale structural variants. And so this would be on the order of 1,000 base pairs or longer differences. And so the authors identified almost 90,000 insertions across these 31 individuals and 35,000 deletions, and then a couple other translocations or inversions or other complex things. And so between any single individual, you're looking at tens of thousands of insertions or deletions and potentially thousands of differences in gene loss or gene gain. And so most of this data would be missed using a traditional short read data, and this is kind of where the field is moving. So something to think about when you're doing these types of analyses is if you're using a single reference and you're aligning to that, you could be missing tens of thousands of genes or transposons that can have really important biological functions. So currently, this is really expensive still, but I would imagine in the next 5 or 10 years, this will become the default, and we need to really have a paradigm shift in the way we think about reference genomes and how we represent them. But this can have really important biological meaning. Okay, so just to summarize what we talked about today, so hopefully after this lesson you have a better understanding of the principles of read alignment and variant detection, so how we go from our individuals of interest, we sequence some Illumina data, we quantify variants, and then what we can do with those. Understand the downstream applications and analyses associated with variant calling. So again, I just kind of skimmed the surface here with a couple examples. You could take whole courses on this. There are tens of thousands of papers, and there are whole fields associated with this. This is really a starting point of getting your list of variants, and there are lots of things that you can do depending on the research questions you have. Importantly, hopefully you learned the distinction between these small and large-scale variants. So the difference between using Illumina data to identify SNPs or small-scale insertions and deletions versus using long-read data to identify these large insertions or deletions or translocations, and the concept of pangenomics. This is really where the field is moving, and genomes can have tens of thousands of large-scale insertions or deletions between individuals. And so the question is, how do we use this data, and what role can it play in the future? And so hopefully this provided you some introduction, and for the rest of this lesson, what we'll be doing is we'll be importing a VCF file, doing some filtering, and doing some basic hierarchical clustering and principal component analysis on some data from grape and some data from pineapple. And again, this is just an introduction. You can do lots of downstream analyses from this as well.
Thank you.